So, welcome to the class of computational biology and bioinformatics. So, today we are going to study sequence variation and polymorphism in DNA sequence. So, this is the same thing. The sequence variation we can include. We can talk about the amino acid variation also. So, your topic is polymorphism in DNA sequence because we know that the source of variation is the DNA and gene, and that is responsible for the prediction of different types of proteins, and which can be visible at the phenotypic level. So, first we study amis. What are the causes behind the variation? Okay, in the population. So first we will start it from the population. What is the population in the Indian population or everywhere at the global level? You can see the variation and differences in the appearance of the people. Also, Negroes people like and uh, the peoples who are fair in color like British people. So you can see the difference variation among the on the level of appearance. Within India also you can see the differences. You can see the variation among the different people. Those who belong to the Tamil Nadu, Chennai, and those who belong to the Himachal Pradesh and Kashmir. So you can easily differentiate among those people. The peoples who the population that belongs to the South India, that belongs to the North India, you can make the difference. I mean, by the appearance. So, what are the causes behind that? So, you can see the differences among vegetables. We have the different kind diversity of the plants. Means we have different kind of fruits, vegetables. That shows there is a variation. Okay, and you can see the differences among the different types of animals also. We have the different kind of animal in the forest like fox, lion, okay, and different types of creatures like snakes and other types of. We can talk about different. Whenever you go visit to the forest, you can easily visualize the differences and variation in the appearance, okay, and in their phenotypic characters or in the structure of the body also you can visualize the difference. That's why you call them the different. You call them by the different names like lion, fox, okay, got, because there is a variation in the structural body also. A snake is different to the like lion, fox, and got. Okay, so there is a visible difference in the structural body also. So in the old time and old period, we used to classify the organisms on the basis of their variation, phenotypic variations, whatever we used to visualize by our eyes. So we can sense the this is the difference. So this is clearly visible among the society also. We have the different kind of plants also. Whenever we visit to the different states of the India, you can easily visualize the difference among the plants and animals, their population. What is the population? Means plants or animals. You can compare the different plants and population of human population also from the different states of the India. So if we talk about the plants, so generally in the different state we used to find out the different kinds of plants. In the south we used to have the coconut tree. So there here we do not have the coconut tree. So there is so much difference. We have diverse kind of plants everywhere. So in the Andaman Nicobar, so we can see there is the flowering variety, flowering that is endemic to that area because they that plant. So we use whenever we visit to anywhere, we can see the different kind of plants and animals. So we can easily differentiate. This is from that state. This is from like we used to have orange from Nagpur. So that is the endemic variety of that. So these are all variation that we can easily visualize these variations. Okay, among the fruits also, whenever you eat different kind of fruits like pumpkin, brinjal. Okay, Shimla mirch. So there are the different capsicum, different kinds of vegetables that you used to eat. So that are not the same. You have the potato, okay. You have the pepper. You have the tomato. So that all looks that all show the variation in the appearance. That is how you are going to identify them, and that also give the clues to for the identification of them. So you identify all the animals, all the organisms on the basis of their appearance. Okay, that is true. Okay, do you want to share anything here? That is true. I mean, you used to identify. I mean, you can easily distinguish between a tomato and potato. Potato is the different vegetable, and tomato is the different. You can distinguish. That is on the basis of variations, because of on the basis of appearance, without doing any dissection, without cutting them, you can visualize. You don't need to cut those things. You don't need to carry out any experiment to identify the vegetables, fruits. Okay, you can easily identify the coconut also. Even you do not grow the coconut here in UP. You just take them from the South India, just okay. So because you can identify those variations. So now in this lecture we will study what are the causes behind them. What means what is responsible for that kind of variation? Can you tell me one cause why we do not have the same vegetable in the different state? We belong to the India. Why we have the different kind of vegetables within different states of the India? Can you tell me? Anyone of you? Can you tell me?
Ma'am, because of different breeds and genetic structure.
Because of
Different
environmental changes.
different genetic
Breeds.
structure, or what did you
Breeds.
breeds, different species.
Yes, ma'am.
Okay. Okay, so we have different species like lion, okay, red, okay, and plant vegetable variety. But that is the okay. But why we cannot grow the orange here? Means why we cannot grow the apple here in UP? Can
Ma'am,
you?
Because of
because of the
different environmental
Because of
conditions.
different
because
climatic situations.
Okay, what's your name?
Ma'am Soumya.
Somya. Okay, you told me that is a climatic variation, no? That is
Yes,
because,
ma'am.
okay, correct. Climates and environment, you can say that climates comes in the
Yes, ma'am.
environment. Because our environment vary. We have different climatic conditions and environment. So environment is responsible for everything. Because in Himachal Pradesh we have the cool atmosphere and Kashmir, so we can grow the apple there. We cannot grow the apple in the UP. So that is the that everything depend on them. Whatever we species variety we have, a different kind of varieties we have on the earth, that is due to the variation in the climate and environment. So environment has the great impact, although it cannot affect immediately. Doesn't show the effect immediately, but it has the, if we suppose we take the plant from here from UP, and we grow that plant, we just transfer translocate that plant into another state. Can that plant can be grown there or not?
No, ma'am.
Okay,
And
why?
it depends on the environmental condition. If the environmental condition is same, then it will grow.
Okay, yes,
Um,
yes. It depends on the environmental condition, that variety, or either according to the Darwin or Lamarck, either it will survive. If it will be best fitted to that new environment, that can survive. There may be the chances of the survival, and there may be the chances of the death also. Okay, both of possibilities. Okay. No, if means the plant can survive in that, and it depends on the what type of soil quality. Soil quality will be needed for that, because we have the different types of soil also in land quality, soil quality in the different states. That's why the variation means the variation among all the physical factors, all the physical elements of the environment. That is soil also, it constitute air also, it constitute water also. Okay, so these all have impact on the different varieties. The water is also responsible for that, the air, atmosphere, that is also responsible for that, and the land is also responsible, the type of land and the soil is also responsible for that. Okay, that you are agree with this?
Yes, ma'am. And light is
Yes,
also.
ma'am.
So sometimes, or if we translocate one variety from suppose abroad to India, so it may show the variation. It can survive there, but there may be the chances that it can be developed into the new variety of the same plant. Like, okay, new variety of the same vegetable if we are growing here, that can show the difference variation in the appearance also if it
Yes, ma'am.
yes, if we have transported that plant or animal from the foreign abroad and we that okay or the animal that is living here, so their next generation may show the variation in the appearance, but it is not very immediate. The animal used to take because in the case of animal their life cycle is long, so it will take time to visualize the differences because the that evolution is carried out over a long time of period.
Adaptive radiation takes place in
Adaptation
the same
also.
place.
So immediate changes, radiation is responsible. So that
Yes,
is,
ma'am.
but that is always harmful. We are not going to talk about this just now. Any immediate change has been resulted into the environmental hazard, just like the population of Japan and Hiroshima after the incidents of the bomb explosion. Okay, so they show the differences also. That it was the immediate, so it has great impact on the population of the Hiroshima and Nagasaki. Okay, those were very sad incidents. So it shows the impact now also. They're carrying out the impact at the genetic level, so that becomes hereditary now. So we were discussing about the if we transport one fruit plant from one state and we can transport it to the another place, so we can grow or not. So it depends on the environment. Environment is responsible. It can show the difference in the appearance. So that all of us we knows that environment is responsible for this kind of appearance. So, but how if there is a change? If there is any change, how it will appear at the phenotypic level. So in the evolution also, so in the plant and bacterial population, we can easily visualize the impact of variation, mutation in the bacteria. Like in the coronavirus, there is a mutated variety. So you can easily see. But in the case of human and animal population, you cannot. You can immediately visualize the differences. If your relatives are have gone to the abroad and they are living there for the one generation, for the next generation also, so if you want to see the impact of the variation, then you need to wait for the few, means more generation for the thousand of years or for the so you are not going to lie, okay, for that much that time period. So evolution takes place. So it is very slow. Environment has the impact, but it doesn't have the immediate impact. Our genetic structure changes with the time, but it takes time. It is not that because plant life is small. Those who have the shorter life period, they are going to change. We can visualize the changes in the bacteria. We can visualize the changes immediately within the few months or within the month. We can visualize the changes, like in the case of the coronavirus. Okay, you can see the changes in the physical appearance of that virus or bacteria. But if you want to observe the changes in the plants or animals, we need to wait. So in the case of annual plant, we can see so that's easy. The organisms which have the shorter life period, we can see the effect of mutation on them. But mutation has same impact on all the organisms. Okay. So we have studied in the genetics like in the study of the DNA that is the variation sequence variation can be one residue per 1,000 nucleotide. But in the case of animals, as I have told you, the human has the longer life, so we will not stay up to that time to see the differences. But the mutation rate is same. So what we vary, we have the different differences in the lifetime period also. What is our life cycle? Our life cycle, and what is the life cycle of bacteria? Because bacteria and virus has the short life cycle. Okay, so we can visualize the changes and the evolutionary changes in those variety. Okay. So environment is responsible for that. These things all are studied in the population genetics. There is one branch that is known as population genetics. In that we study the about the different species and what are the changes that comes changes at the gene level that can impact. So Mandel, on the basis of phenotypic appearance also we can study the changes. So it has been started from the Mendelian genetics. The Mendel has done so many experiment. He used the plant Pisum sativum for the experiment, produce the different kind, crosses the different varieties of the Pisum sativum. Okay, that is yellow, green and yellow, round and wrinkled. So you already know about that. You knew that. Okay. So what were the differences? Okay. So it has been started. The population genetics. It has been started from the Mandels. So Mandel has done the experiment, and then was the development of the genetics branch. So if we are analyzing the different types of population, then it is the population genetics. So it has been started. Nowadays, we also do carry out the DNA study because we have done so much advancement in the technology. So we are going. So this is the because we have done so much advancement in the technology. So the success of the human genome sequencing program. So this has stimulated the interest to work at the genetic level because now we have the sequence. In the old time, in the old period, didn't have the sequence or DNA to compare all that. So in the population genetics also, we used to study the phenotypic on the basis of phenotypic appearance. That what the Mendel has done. After that, Hugo de Vries and other scientists have done on the basis of physical appearance. And we used to classify the organism also on the basis of the phenotypic appearance or the biochemical character. Okay. Whatever means it depends on the time. I mean generation by generation or by century. Okay, different centuries we have done so much advancement in the technology. So on the basis of that, we were characterizing the organism. We were classifying the organism in the hierarchy. You know, the class, phylum, genus, order, species. But now we used to classify. We use the DNA sequence to classify we used to compare the protein and amino acid sequences. After the so many invention, the biotechnology and we have the sequence. We got the sequences in the biotechnology. We got the sequences, and then the bioinformatics field appears. So using the bioinformatics approaches, we have compared the sequences using sequence alignment method. So those methods nowadays are helpful to characterize the proteins and gene family on the basis of proteins and sequence. Sequence alignment methods are used that can give the important clue to characterize important bases to characterize the genes and proteins into families. Okay, So now we will start from what are the important methods. Okay. So this is just introductory part. So due to the advancement in the methods, now we have a lot of new definitions. Now we can characterize the gene and protein at sequence level. We are able to characterize them because of the advancement in the technology. Now we have the new method. So there are some new definitions. What we have discussed just before. Now we have some new definitions also because of the biotechnological and bioinformatics advancement in the technology. So human balance, especially those that are relevant to the disease, their treatment and their cure. We are interested. Why we are always interested to study the DNA and protein that is related to disease? Because all of us we know that we fall sick and we visit to the doctor. Sometimes there is a complicated disease that cannot be characterized treated easily. So daily we are doing the means nowadays we are doing the advancement in the medical technology also or biotechnological experiment that can help us to find out to diagnose the causes of the disease also, to diagnose the disease in the human, and to see the what are the fundamental causes behind these types of disease. Because now we want to know at the DNA level, because we have the DNA sequence, so we want to know what is happening there, why this type of disease are occurring in the population or in our human population. So even nowadays we are also characterizing the plant diseases on that level, plants, because that are also important to us because we are dependent on them. Plants are the producer and we all dependent on them for our food requirement, food resources, for the fresh air also, because plants are the necessary component of our environment. These are the producers that provide us the fresh oxygen, fresh environment. In that, from that atmosphere we inhale the oxygen from the atmosphere, then we exhale the carbon dioxide. So that's so using the reaction of photosynthesis, plants synthesizing the different types of foods in the presence of chlorophyll and sunlight. So we are dependent on that type of food, so humans are dependent on them. So we are also careful uh, in case of plant disease also. Plants should not have any disease because we want to eat them. We eat the product of those plants. So that is also going to impact the human. If we eat the bad food or the disease plants, so we can have the disease. We can develop the fungal disease or anything, any disorder. So these all affects. So we are interested because of our means medical problems, because we don't want to have any disease. We don't welcome any disease. So what are the causes behind them? So first we are using, there is a means genetics methods in the medical field that is relevant to the disease, that is important to us. Okay. So this requires the use of more genetic markers, and the scientists are discovering that also. So in the previous class also we have discussed about the genetic markers. I have given you the study material regarding that also. Genetic markers, molecular markers. Okay, that can identify the presence of gene. So that markers work as a landmark to identify for the identification of the gene. And gene mapping studies played an important role. So about the gene mapping, we have already discussed. There are the different types of gene mapping methods like genetic mapping, physical mapping. Genetic mapping means that is based on the assuming distance that was given by the Morgan distance, anti-Morgans. Or that is based on the some kind of experiment like crossing the population, like how many recombinants are there, how many pure population is there. That is some kind of Mendelian experiment. So on the basis of those cross population, pure population, we can calculate the recombination frequency using the and then that provide the clues about the linkage and crossing over among that type of plant population. So generally in plant breeding, whatever we do in the plant breeding, okay, that is related to that. So the use of genetic markers. So in the case of plant, we are doing more experiment and we are doing more advancement because that is very helpful for us also in we have already discussed how those experiment how the type of knowledge is used in the crop improvement. You have a previous class on the crop improvement. So how we have used that information in the improvement of the crop and the prediction of the hybrid variety without changing and without mutating any gene sequence. So we used to produce the different types of plant hybrid varieties using the old population, using the pure variety, wild varieties, and we are producing the hybrid varieties. As you see, you can see the case of the tomato also. Now you have the improved variety. Okay. So the more experiment we are doing on the plant because it is easy to do that. So, and it is also advisable to eat the healthy food to stay away from the disease. So that is the if we eat the good food, we will not have any disease. Somehow, if we get the disease, we are providing solution for that also. So we are interested in the genetic studies that is related to the medical field. Also, although we want to avoid that, but sometimes we get the disease. You can see if you visit to the hospital, you can see so many patients are there. They're suffering from the diseases. So these are the two interests that we have the motivation work on the plants or animals also, or especially for the human because we are the humans, so we want to stay away from disease. So there is so much advancement. The international consortium of 10 pharmaceutical companies. So because they are all working together. Okay, and Wellcome Trust has taken up the task of finding three like the means 3 lakh random single nucleotide polymorphism spread throughout the genome. So the companies, pharmaceutical companies, they are working together to work on these single nucleotide polymorphism that spread throughout the genome. Okay, because these SNPs are maybe related to the human disease. So we have interest. We want to know what are the causes of different types of disease. What are these SNP variations? So all the pharmaceutical companies are working together and with the help of Wellcome Trust. Okay. National Institute of Health and some commercial they also concerned about they have also initiated their own effort to collect the large number of random SNPs. So what is SNP? Single nucleotide polymorphism. You already knew about this because previously we have also discussed about the single nucleotide polymorphism. There is a single mutation that you can remember the case of sickle cell anemia. Okay, so in the sickle cell anemia, there is only single mutation that is responsible for the production of the sickle shaped RBCs. That RBCs, sickle cell RBCs, are not able to carry out the more oxygen. The oxygen carrying capacity is less in that case. What the normal RBCs have. Normal RBCs have red blood corpuscles have more oxygen carrying capacity. So you can see how the SNP can have impact on the function of the proteins. So second school of thought was that SNP should be mined in coding and regulatory regions. So what are the causes in the coding regions? The mutation happen in the case of the RBCs also. The hemoglobin has a mutation in the coding region. If the mutation, I mean generally in the nature, why it is written like that? Because generally in the nature, the impact of the mutation is always minimized. It goes in the introns. Generally, the mutations are found in the intron region. But sometimes it can appear in the coding region and regulatory region. So this has the great impact. And this is responsible for the manifestation of genetic disorders, which would point more directly to disease causing mutation. Studies could then focus directly on candidate genes. So we are always interested to know what are the disease causing mutations. So studies could then focus directly on the candidate gene. So we can, if we are able to identify that gene, we can work on that. Well, how we can minimize the effect of that mutations. So generally the mutation goes in the intron region. So one example of that is we all have junk DNA. Our means 95% DNA junk DNA. So that is of intron. They consist of totally of introns. But the intronic DNA is important because we have, that is our ancestral DNA. Ancestral DNA. And that vary with among all the humans. On the basis of that DNA, we identify the parents of offspring, like in the DNA fingerprinting. So that DNA have the different kind of pattern, like VNTR, variable number of tandem repeats, mini satellite, microsatellite variations. So there are number of repeats. So that patterns vary among all the individuals, among all the human population, all the individuals of the same population also. So on the basis of that, the father-son relationship can be detected in the DNA fingerprinting. So that is the another case. But our means less than 5% DNA is coding DNA. If mutation appear in that region, it greatly impact our uh, our protein production. So that can be manifested at the disease as the disease, because that mutation will go in the at the protein level. The protein is functional in our body, and that if there is an important enzyme, that will be manifested in the at the phenotypic level also. So whatever the approach, the task of finding candidate gene by genetic method require large scale analysis. So it needs, although because the human genome is large, also the task of so sequencing and resequencing. Resequence is written here. Because why we do the resequencing? Because all the individuals can show the variation. So resequencing may be required. We say that the human DNA is the same, the human genome is the same, but coding DNA will be the same. So in candidate gene may also be a large one. So we can take the individual from the different types of population, like Caucasian population or Negroes population. So once a disease associated gene has been discovered, the spectrum of the mutation characterized. So what they are talking about, disease associated gene. Disease associated gene, we should try to identify that gene. So, but that is very difficult. That we should, but now because of the advancement in the technology, now it is facilitated. We can identify the gene from the different types of population or the person that has the disease. So that study is costly, but we because we are interested to know, we want to diagnose properly, because if we want the treatment proper and cure, if you want to cure the disease, that is always of great interest. So there may be need to test the large numbers of individuals who are at the risk for the presence of these mutations. Okay, so if we test the large number of the individuals who are at the risk Because of these types of mutation that may provide the important clues about the functioning. I mean, how the DNA is responsible for the dysfunctional proteins or misfunctional proteins. Because if that mutation is at the DNA level and DNA produce the protein, so we have discussed already in the open reading framework how the mutation, point mutation have impact on the prediction of the protein. So SNPs has the great impact on that. Even we have the single mutation can cause the serious disease like sickle cell anemia. So there is a need for large scale analysis of sequence variation at the first stages in characterization of human trait. Trait means large scale analysis. Obviously, we need the large scale analysis of sequence variation in the characterization of the human traits. What are the human traits? Means like the height is the one trait and height is the one character. What are the two different traits of the that height, that is dwarf and tallness? These are the two different traits of the same character. Okay. So in the case of all the proteins and in case of all the, and then there will be the normal character. There may be the different traits. Like in the sickle cell anemia, we have the two. One is normal and another one is a sickle cell type of trait. So human trait means the person who has the defective genes and the person who has the normal gene. So that will be the two traits. So there may be some another traits also additional to that. So this will provide the informative sequence variants. So we have the different, if we have the different kind of sequences in the database, so we can map and mapping by the association or linkage. So that is a method, genetic mapping. We do that. We use to map the genes. Okay, in the genetic mapping, mutation analysis in the candidate genes and diagnosis in the individual. So these are the four different levels. On these levels, we can identify. We can have the different sequence variant because we will discuss. This information is available in the NCBI database in DB SNP. Like sequence variants, we can find out.
एक्सक्यूज मी मैम
Yes.
मैम हमारी दूसरी क्लास का लिंक आ गया है
Okay. I'm just going to close this. Okay. So diagnosis in the user, analysis of sequence variation has application in field other than the human disease also. Means other than the human, we also use these types of studies in the case of plants also. Okay. So this need to be done at the large scale. So today we have studied about the introduction of the variation. So on the next turn, we will discuss about the SNPs. What are the different definitions related to sequence variations? Okay. So this was the reference of this class in this. I have taken the material from this, but today's whatever we have discussed today, that was the general information. General I have discussed here. Okay. So we. Okay, these are the students who are present in the class. I already took your attendance.
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Thank you, ma'am.
थैंक यू मैम
Thank you, ma'am.
Okay. Now you can leave.
धन्यवाद मैम.
Now you can leave the class. Okay.
Okay, ma'am.
ओके मैम
Okay, ma'am.